Good day, everyone. My name is Asli Heitzer, and I am the Program Lead for HIV Prevention Research and Development at the Global HIV Vaccine Enterprise of the International AIDS Society. And today it is my big pleasure to open this timely topics webinar on recent advances in eliciting broadly neutralizing antibodies for HIV vaccine development. I would like to welcome all participants as well as our speakers and moderators. And today, this webinar aims to explore cutting-edge strategies in HIV vaccine development and also highlight recent advances in immunogen design and vaccine platforms that guide immune responses toward broadly neutralizing antibody development. Today's meeting is comprised of 6 presentations, followed by a 20-minute Q&A, and we encourage all our participants to use the Q&A function on Zoom during the presentations to submit any questions you may have. I also would like to highlight that all of today's presenters have co-authored a newly published article in the Journal of the International AIDS Society with the title Status of HIV Vaccine Development, Progress and Promise. This article provides an overview of the current landscape of HIV vaccine research, and we will post the link in the chat, and we really encourage you to click it and read it. So some housekeeping rules. Please note that this meeting is being recorded, and by participating, you consent to be recorded and also for the recording to be used publicly. Having said that, I would like to now introduce our moderator and presenter for this webinar, Barton Haynes, who is the Frederick M. Haynes Professor of Medicine and Immunology and Director of the Duke Human Vaccine Institute. Barton has led 3 major NIH-funded consortia, CHAVI, CHAVI-ID, and CHAVD, focused on understanding HIV transmission and novel HIV vaccine immunogen design, and he will give us an overview of HIV vaccine strategies. Bart, the floor is yours.
Thank you so much, Asli, and thanks so much for the opportunity to have this webinar today. And it's in honor of the HIV Vaccine Awareness Day, which occurs in 2 days on May 18th. So this is a very timely topic for all of us. So for conflict of interest disclosures, the team that's presenting today has patents on immunogens being discussed, and the speakers are all members of the NIAID Division of AIDS-funded Duke Consortia for HIV/AIDS vaccine development, called CHAVD. And we also have grants from the NIH and the Gates Foundation. So the first question is, why do we need an HIV vaccine? Well, in spite of antiretroviral therapy taken as pre-exposure prophylaxis, or PrEP, unfortunately in 2023, approximately 1.3 million people worldwide newly acquired HIV. And so we need an array of HIV prevention tools accessible to everyone, and of that, an HIV vaccine would be indispensable to end the pandemic. And so vaccines have been the most effective means to prevent and eradicate infectious disease. And as I said, a preventive HIV vaccine could well save millions of lives. So why are we here 20 years after the start of the CHAVI series of grants programs, and we don't have an HIV vaccine to deploy? Well, several reasons. The first of which is that none of the 9 HIV vaccine efficacy trials from 1999 to 2019 showed any protection, except what's called the RV144 trial that was carried out by the Military HIV Research Program in Thailand. The RV144 trial was follow-up trials in Africa, however, showed no efficacy. And all 9 trials induced what's called cytotoxic T cells or killer T cells to kill virus-infected cells, or induce non-neutralizing antibodies. But there was no induction of a type of antibody we call broadly neutralizing antibodies. So the field has realized that we need to induce these rare antibodies called broadly neutralizing antibodies, and the rest of the webinar will call them bNAbs. So bNAbs are antibodies that neutralize HIV by binding to conserved regions on the HIV envelope on the virus surface. There are 8 such envelope regions, and we call these epitopes. We'll need to induce at least 3 to 4 specificities of broad neutralizing antibodies to prevent HIV mutation and prevent their escape from vaccine-induced bNAbs, and also to cover the broad range of HIV variants. So in essence, we're essentially having to develop 3 or 4 separate vaccines at the same time and combine them together. So this is an illustration of the HIV envelope here in the middle of the screen, and the different colors show the different epitopes or conserved binding regions of broadly neutralizing antibodies, and there are 8 of them that have been identified by the field. And today we're going to talk about the 4 most potent of these epitopes to which broad neutralizing antibodies bind. We're going to talk about the V3 glycan, the V2 apex up at the top, in teal, the CD4 binding site, and down below in gray, the membrane proximal external region down near the virus membrane. So broadly neutralizing antibodies are disfavored by the immune system. This is another reason why we don't have a vaccine yet. And that is the immune system fundamentally doesn't choose to make these kinds of antibodies at the beginning of the immune response. Potent broadly neutralizing antibodies actually rarely occur in people living with HIV, and bNAbs take years to develop in the setting of HIV infection in people living with HIV. bNAbs are very unusual antibodies. They have long antibody combining sites. They have unusual shapes. They have many rare and probable mutations, and they're can react with other kinds of things, fooling the immune system. All these are traits that can lead to the immune system not wanting to make the antibodies. And the bNAb binding sites can be very complex. They're either combinations of proteins and sugars or glycans, or proteins and virion lipids. So what's our long-term goal in this CHOV-D program? Is to develop a vaccine that targets 3 to 4 bNAb binding sites, that induces durable serum 80% inhibitory titers of at least 1 to 100 or more, and a combined neutralization breadth of greater than 90% of HIV isolates. So what's the solution? We think the way to induce broad neutralizing antibodies is to design immunogens that target the bNAb B cell receptor. Down here, this black antibody representing a naive B cell receptor, and we design an immunogen in purple that will target it. These are called naive B cell receptors or germline receptors. By presenting a progression of immunogens, we can guide naive B cell precursors to potent bNAbs by these sequential orange, green, and blue representation of immunogens there, and the broad neutralizing antibody is in dark red. And we believe that iteration in small vaccine trials, which Lindsay Baden will talk about, is key to optimizing each sequential immunogen. And the HIV Vaccine Trials Network, funded by the NIAID, calls these small trials discovery medicine trials. So how far have we come towards the solution? Well, CHOV-D programs can induce bNAb precursors for the EMPER down at the base of the envelope, CD4 binding site in teal, and V3 glycan, and have done so in humans. And you'll hear a bit about those studies in a moment. We've also isolated proximal EMPER bNAbs that neutralize heterologous HIV isolates from humans that neutralize 63% of the clade B that's common in the United States and Western Europe, and 19% of global isolates. So we have a ways to go there, but it's a beachhead. We have achieved induction of serum HIV bNAb breadth for V2 apex bNAbs in rhesus macaques, and Beatrice Hahn and George Shaw will talk about those data. Our remaining tasks are to design boosts that finish the maturation of bNAb development and get to the end so that they're potent enough and broad enough to be clinically successful. And we have to learn to induce long-lasting high titers of broad neutralizing antibodies, because unlike other vaccines, at the time of potential transmission, the titers have to already be high enough to be protective. And then we have to formulate this complex series of immunogens as a practical vaccine. So all of the work that I just referred to and you will hear about today is funded by the NIAID, Division of AIDS, and through 3 grants. First one was the CHAVI in 2025, the second one in 2012, the Center for HIV/AIDS Vaccine Immunology and Immunogen Discovery, and the current grant, the Consortium of HIV/AIDS Vaccine Development. And we're in partnership with the HIV Vaccine Trials Network that Larry Corey leads and Lindsay Baden is a member of, that performs all the clinical trials. So without further ado, I will now introduce George Shaw, our next speaker. Dr. Shaw is Professor of Medicine and Microbiology at the University of Pennsylvania. And George is going to talk about some exciting new data on the use of simian human immunodeficiency virus. Let me stop sharing for George. Simian human immunodeficiency virus animal models as a new and very powerful model of broadly neutralizing antibody development. We've never had an animal model like this, and he will tell us how it's informing HIV vaccine design. George, please take it away.
Thank you, Bart, and good day, everyone. The title of my talk is SHIV Models of Broadly Neutralizing Antibody Development. And our premise is that an outbred animal model can play a critical role in the development of an HIV vaccine for humans. And that's because an outbred animal model can allow us to discover new immunogens that can elicit bNAbs, and it can allow us to iterate trials so that we can develop those study after study in order to develop what we think will work in humans. And this is a complement to the human studies, and it is a strategy that can work in parallel with human studies. And you'll see that in my slides today. So I work on SHIVs. SHIVs are simian-human immunodeficiency viruses that essentially take a naturally occurring monkey virus that has the same structure as HIV, the same genomic organization, but this virus is not sufficient for an outbred animal model because it contains a monkey envelope. And what a SHIV is the HIV-1 envelope substituted into the backbone of a monkey virus. So SHIVs and HIVs exhibit similar replication dynamics in vivo. HIV in humans, SHIVs in monkeys. And you can see that here, viral RNA copies per ml on the y-axis, time in years on the x-axis, and both SHIVs and HIVs have characteristic viral replication dynamics. They also have similar dynamics in the elicitation of HIV binding antibodies. These binding antibodies are what turn your ELISA positive, your Western blot positive, and allow for an antibody-based diagnosis of HIV. SHIVs and HIVs also elicit similar autologous neutralizing antibody responses. In other words, the B cell response to HIV in humans, the B cell response to SHIV infection in monkeys is very similar. And you can see here in the red line that essentially 100% of humans infected with HIV-1 and monkeys infected with SHIVs develop strong autologous neutralizing antibody titers with these kinetics. They occur just weeks after seroconversion and infection with HIV. Now, what we are aiming to get here in this talk is get to the bottom of how do you develop broadly neutralizing antibodies. And the way people have done this in the past is to look at the natural human infection. And what you see in human infection is not weeks, but rather years after infection by HIV-1, you see the development of heterologous or broadly neutralizing antibodies, but only in less than 10% of people. So these are rare antibodies that are uncommon. And of course, what we want in a vaccine is heterologous neutralizing antibodies elicited in 100% of people. So we think we need an outbred model in monkeys, in this case, to develop immunogens that can change less than 10% to greater than 60, and then greater than 90, and hopefully in 100%. And so this dark blue line here is what I'm going to talk to you about today. I said at the beginning that there is no outbred model for the elicitation of broadly neutralizing antibodies. And what I'll show you today is that there now is one. And this outbred model is SHIV infection, and it elicits broadly neutralizing antibodies greater than 60% of monkeys, and oftentimes greater than 90%, against the V2 apex, canonical bNAb site, the V3 glycan site, the CD4 binding site, and the fusion peptide. And that's my message to you today. So how do we do this for V3? Well, there's been a history by Steichen and Schief and Escalano of developing germline targeted sequential immunogens aimed at eliciting V3 glycan bNAbs. And what they started with was BG505, did mammalian display mutagenesis, and developed different variants that were found to bind to inferred germlines of V3 glycans. The problem is they work great in pre-rearranged VDJ knock-in mice, but they don't work in outbred animals. 10 years into the work, no bNAbs. We focused on one of these envelopes, SHIV5 mut. We reconstructed it into a SHIV, and we infected monkeys. Now, if we had done this with just regular old BG505, the wild type strain, we did do that, in fact, and we found in 0 of 14 monkeys bNAbs. In contrast, in 22 monkeys infected with SHIV5 mut, we found bNAbs. 14 of 22 monkeys, 64%. This is transformational. This is what the fields look like for the last 20 years. This is what the field looks like now. And here is just a set of those 22 monkeys, and you can see that most monkeys are developing broadly neutralizing antibodies. These are reciprocal ID50s of plasma taken from each of these monkeys here. And you can see that the majority of monkeys, look at this guy here, AM12, he's neutralizing all 8 out of 8 or 9 out of 9 heterologous viruses in titers of 1 to 5,000, 1 to 2,000. And this neutralization shown here against a heterologous virus Q23 and its N332 knockout is strictly N332 dependent. That's the diagnosis. That is the feature of V3 glycan bNAbs. We cloned monoclonal antibodies from each of these monkeys. They're shown here, and the monoclonal antibodies potently neutralize heterologous viruses. And at the bottom here are the immunogenetics of all these monoclonal antibodies. And the take-home message here is that the HCDR3 length ranges from 19 to 25. And most recently, 18 to 25. Most recently, we found 3 more monoclonals with HCDR3 lengths of 14 and 15. What that means is that these types of antibodies are prevalent in monkeys, and they're undoubtedly prevalent in humans. The V3 glycan bNAbs that we've elicited, this is one here, AM12. The structural interactions, the paratope epitope interactions with the envelope are virtually indistinguishable from a human V3 glycan bNAb. And that's true for all 10 bNAbs we've identified. They look just like humans. I'm going to switch gears now to the V2 apex. It's a different bNAb target. It's a bNAb target that sits at the apex of the virus. And the Duke Chavit has developed an immunogen called OPT4 that you'll hear about in the next talk. You'll hear how this, as a protein nanoparticle or mRNA, elicits V2 apex bNAbs. I'll show you here what it does when you construct this piece of V1, V2 into the Cap256 ONF backbone and put that into a SHIV. There it is in the SHIV. Here are the replication dynamics of the SHIV. Here are the broadly neutralizing antibodies that are elicited in the first animal. So on the column here, this is the animal, CI93. This is weeks post-infection. This is the neutralization of the plasma from these weeks against MULV. This is against the autologous virus. The money on this slide is right here. By week 20, we have heterologous, broadly neutralizing antibodies neutralizing T250, ZM233, Q23, CAM13, H703 in titers between 20 and 1300. And these reactivities are strictly C-strand dependent. This C-strand dependency means they are absolutely diagnostically V2 apex bNAbs. So what I've just shown you is that we can elicit bNAbs against the V3 glycan epitope, the V2 apex epitope, and this is one monkey, but we have 16 more. If you ask what is primed in these monkeys, what are these antibodies? Here are 6 different lineages in the first 16 weeks of infection, and the money is right here. HCDR3 lengths of 23 to 25, and the D gene is this D3-9, also known as D3-15. These are diagnostically, unequivocally, V2 apex bNAbs. I'll conclude with this slide showing you that we've also developed a SHIV model that will elicit CD4 binding site bNAbs. Here is an illustration of 115 monkeys that we infected with SHIVs. And we found out of 115 monkeys infected with natural SHIVs, with natural OMPs, one monkey with CD4 binding site bNAbs. We genetically engineered the envelope to lack glycans at 3 positions, 461, 362, 197, surrounding the CD4 binding site. And we go from 1 in 115 monkeys with bNAbs to 9 of 11. And finally, we've done the same thing with fusion peptide. And I'll just conclude by pointing you to the Cell reports. This paper is in press. I'll point you to Cell 2024. This paper is published in this citation here in Cell. My conclusions are the following. Designer SHIVs expressing novel germline targeted OMPs can markedly enhance bNAb elicitation in rhesus macaques and serve as a molecular guide or blueprint for HIV-1 immunogen design. We've developed new SHIV animal models to elicit V3 glycan, V2 apex, CD4 binding site, and infusion peptide bNAbs. And the following presentation shows translation of SHIV bNAb elicitation into novel protein and mRNA designs for the elicitation of the same V2 apex bNAbs. People in my lab who've done the work are highlighted here, a great group of people, and I'll end.
Thank you, George. Very exciting and great talk. And hopefully there'll be some questions about how this marvelous model can be used. Our next speaker is Beatrice Hahn. Dr. Hahn is professor of medicine and microbiology, also at the University of Pennsylvania, Perelman School of Medicine. And Dr. Hahn is going to talk about, as George alluded to, targeting the V2 apex now with a vaccine, a series of candidates, and the success there in non-human primates. And then also, I believe, we'll mention an upcoming clinical trial. So Beatrice, take it away.
Thank you, Bart, and welcome to everybody. So our target is the V2 apex, as has been said before. And the V2 apex antibodies have some unique features. They bind, as the name says, to the tip of the trimer. They need a real trimer to bind, and they have to navigate what is shown here in green, glycans that stick up in order to find their epitope. And for that, they need a very long, heavy chain third complementarity region to reach between those glycans right down to the protein backbone to hit the epitope. And the epitope is also called the C strand because from the structure, it's called the C strand, and it's position 166 to 131. So when you hear about C strand mutants, they are introduced to knock off the binding of such antibodies. In people living with HIV, they have been isolated and they have good neutralization breadth and potency in the people that actually develop them. And they represent the second most frequent bNAb specificity in infected humans. And so we hope that we will be able to elicit them with a vaccination. So George has talked about OPT4 as our priming immunogen for V2 apex bNAbs. Our group is using a very different approach from others, and that's primarily Betty Korber and Shidit Chua. They use neutralization signatures to design a priming immunogen. And it's important for you to understand that all I'm talking about here is a 90 amino acid stretch of V1 and V2 at the apex that we have modified. And the modifications include, we've taken out a glycan at 130, which is a known hindrance to bNAb development. we've exchanged the V1 and the V2 loops with those of naturally occurring strains that are particularly short and particularly positively charged. And then we have modified the C strand again. That is the protein epitope that the antibody binds to. We tested this in several different envelope backgrounds, and the one that works the best is Cap256 from a person who actually developed a V2 Apex bNAb from South Africa. And the way we determine what is best is we ask how many human and macaque V2 Apex bNAb precursors are actually binding to that. And so this constellation was the best. We also changed one amino acid, which then introduces a glycan in this region here, which we call the V2 prime. And we did this because we realized this is a glycan sparse region that will probably be immunogenic and elicit antibodies that we don't necessarily want. So we then went on and delivered these designs as mRNA immunogens expressing stabilized membrane-bound ONF trimers. And so we went into monkeys, 12 monkeys to be specific. We immunized them with 200 micrograms of mRNA LNPs, 50 microgram in each limb. And these immunizations were given at 10-week intervals. And you can see here, we started with OPT4, and then we did immunofocusing with this OPT4 shielded immunogen that I just described. So what did we get? The good news was that the OPT4 immunizations elicited high titer autologous neutralizing responses. So OPT4 is highly immunogenic, and the plasma ID50 titers reached hundreds of thousands in each animal. So that's good. But as we had suspected, most of the induced neutralizing antibody responses mapped to this glycan devoid region in the hypervariable V2 loop. So we went in with 2 additional shielded OPT4 immunizations, and that not only improved the autologous NAP titer in all animals, but also increased the fraction of antibodies that recognized the C strand and are epitope specific. And in fact, 3 monkeys developed very limited low titer plasma breadths. So that was a good sign. And then we in the Duke group did a number of B cell cloning experiments, pulling out these B cells. I don't have time to show you this, but what we pulled out of the various time points is antibodies that had the right features. They had the long HCDR3 loop that you heard about, and they neutralized the autologous virus in a C strand and N160 dependent manner. So they were really targeting the right epitope. And so the bottom line is this OPT4 shielded regimen is a fantastic priming immunogen, and that worked. So then we said, okay, what are we going to do next? We have primed and it seemed to have worked, but now we have to boost. We have to boost these lineages that we elicited along the desired pathway so that at the end, hopefully we get bNAbs. And so our approach, again, is a little bit different from others. We try to select the heterologous boost in real time. And that means after the priming and immunofocusing, we extracted IgG from the plasma of these immunized macaques and asked, can that plasma IgG bind to a set of envelopes that again, Shidich and Betty had selected based on the signature analysis. Which ones of these, we had a set of about 15, which of these can this IgG bind to in a C-strand dependent fashion? And long story short, we selected Q23, gave that twice, then we selected C1080, then we selected 2 additional envelopes, and ultimately ended up with BF1266. Importantly, these heterologous boosts did not have an N130. I told you this is a big stumbling block for bNAb development, but by this time, these envelopes do have an N130 because we had to introduce it to train the lineages that we had elicited and partially matured to recognize N130. So what did we get? And importantly, these are all native ONFs. There's nothing artificial about them. They come from the selection of diversity that's out there for HIV-1. So what did we get? Well, this is the money slide. Let me explain what's on this rather complicated slide. Here are your 12 monkeys, and the colors indicate reciprocal plasma ID50s. So lower titers are in green, medium titers are in orange and yellow, and then the red titers are high titers. We show the plasma neutralization phenotype after each immunization. So the first Q23 boost, the second C1080, and so on. And then we show neutralization to these viruses here that have different colors. The color blue we don't count because these are immunogens. So, you know, if you immunize with something and you get neutralizing antibodies to it, that's considered autologous. So we don't count them. But the ones in red are truly different tier 2 heterologous arms that have been neutralized by the plasmas of these monkeys. And they are divided into N130 lacking and N130 containing. And you can see that the initial boosts that we did, you know, that we got some cross-neutralization to N130 lacking viruses, but right up to the last boost, we got nothing what the N130 containing viruses concerned. But the last boost was an N130 containing envelope, BF1266, really improved that and now elicited more breadth and potency for the N130 lacking, but also for the N130 containing viruses. And so 8 out of 12 rhesus macaques developed heterologous plasma breadths. And that is what you need to be able to induce if you want to have a protective vaccine. The titers in most animals are not sufficiently high yet to be protective. We know what the titers are supposed to be. They are supposed to be about 1 to 200 to 1 to 400, depending. And in most animals, we have not yet achieved that. But in some animals, we have. And there are in particular 2 animals where the titers are really skyrocketing. 41,000, 101,000. Even the N130 titers, which are lower, we have in this monkey and also in this monkey. And in fact, 5 rhesus macaques neutralized heterologous virus CH505 here, and here, which has a glycan at position 130. And the reason why I point this out is this is actually a challenge stock, well characterized, that we have in the freezer. So should we be able to boost these titers up even more after the next 1 or 2 boosts, we will be able to actually challenge these monkeys and see whether the antibody responses that we have elicited are protective. Obviously, we did a lot of B-cell cloning here too, and we pulled out monoclonal antibodies that recapitulated the plasma breadth. So the last thing that's important is that Kevin Saunders, who will be speaking after me, said, okay, can this be reproduced? Is this a one-trick pony, or can we actually recapitulate this in a different trial? And so instead of using mRNA, he used opt-4 ferritin nanoparticles, beautiful nanoparticles, which now bound the great majority of all the UCAs that we have available because of the increased avidity of the nanoparticles. And he immunized 5 rhesus macaques with opt-4 nanoparticles. And then this is the plasma neutralization phenotype showing you. And you can see after the first immunization, only opt4 was neutralized, but after the second, we saw increasing titers, also titers to the shielded version. And then this one monkey developed heterologous breadth, hadn't seen Q23 yet had antibody titers of over 400. And then Kevin boosted these monkeys with the shielded mRNA, and this is what the plasma neutralization looks after that shielded boost. The initial monkey, again, now has heterologous breadth in the plasma, and most recently, Kevin and his colleagues have B-cell cloned, and the clones we pulled out from this monkey and from this monkey have heterologous breadth. So this clearly recapitulated what I just showed you for mRNA, but not only did it do that, it also accelerated the elicitation of bNAbs by using a protein immunogen. So Bart has made the two mRNA immunogens, the OPT4 and the OPT4 shielded, so far as mRNAs, but he's also going to do the proteins, and human trials are being planned for the next year. So in conclusion, the OPT4 and OPT4 shielded priming and immunofocusing strategy consistently stimulated and expanded authentic V2-Apex bNAb precursors in the rhesus macaques. Authentic means they don't only look like precursors, they actually can be matured to become bNAbs. The heterologous boosting that we used was the native ONF trimers, did this maturation, and we matured these expanded lineages along desired pathways, and we induced plasma breadth in 8 out of 12 rhesus macaques, including N130 containing viruses, which is a big hurdle. And so the consistent precursor stimulation and elicitation of plasma breadths in an outbred animal model across 3 different immunization platforms, I'm including here the mRNA, the nanoparticles, and the SHIV model, really represents an important vaccine milestone. This works. And the 2 clinical trials that are planned for the next year hopefully will recapitulate this, at least the priming stage in humans. These are the people that helped, and of course, our funders.
Well, just terrific, Bea, and very, very exciting. And just a comment about the clinical trials, the vaccines have already been made as mRNAs. They are made, and what's called the INDs, what goes to the FDA, are being prepared right now. And the clinical trial, we hope, will start this summer. And so one theme you'll hear throughout all these talks now is the theme of translation of the discoveries and acceleration of the progress that we're making and as a field. And at Duke and the Duke CHOP-D, we're able to do this translation quickly because we built a vaccine facility at Duke University, and we just have to walk down a floor and work with our vaccine team to get this done. Now, the next speaker is Kevin Weehee, who is the Norman Lettvin Associate Professor of Medicine at Duke University. He's Director of Research and Director of Computational Biology in the Vaccine Institute there. And Kevin is going to talk about the next target for bNAbs, and that's called V3 glycan bNAb induction. Kevin, take it away.
Thanks, Bart. So today I'm going to present our work on a component of our overall vaccine development strategy called mutation-guided vaccine design. And I'm going to show results applying this strategy for inducing V3 glycan broadly neutralizing antibodies. So one of the guiding principles of HIV vaccine development that's been adopted by many in the field is to learn how bNAbs are induced in natural infection and then apply those lessons to develop vaccine design strategies. So as a reminder with what we're dealing with, bNAb lineages arise first from very low numbers of progenitor B cells in the naive repertoire, and those then embark on very long and torturous evolutionary journeys where they seldomly mature fully to become bNAbs. And HIV bNAbs tend to have extraordinary numbers of mutations relative to conventional antibodies, and in particular, they have many mutations that are not typically made by the somatic hypermutation machinery, and we'll get into that in a moment. So our challenge is to recapitulate this extremely difficult and inefficient process in infection with a vaccine and to do it reliably. And the way we and many others have tackled this challenge is to break this up into 2 pieces. The first is priming strategies like germline targeting to activate the rare B cell precursors with the properties at the outset that give them a leg up on becoming bNAbs, and then boosting strategies that guide their maturation uphill and against the odds to become bNAbs. We've seen some great progress with germline targeting, but there's been less progress that's been made in boosting. So this talk is going to focus on a new boosting strategy, which we call mutation-guided vaccine design. So the foundational principle on which the mutation-guided vaccine design strategy is based on the statement that not all antibody mutations are created equally. And because of biases in the somatic hypermutation machinery, mutational targeting is not evenly distributed across the entire antibody sequence. There's positions of high mutability, there's positions of low mutability. So this means that amino acid substitutions during affinity maturation are not equally likely. And we term antibody mutations that are not routinely made as improbable mutations. And we've developed a computational tool called Armadillo to estimate mutation probabilities, which we can use then to identify the improbable mutations. And I just want to put in a quick plug that we've got a web server now for Armadillo where you can put in your favorite antibody sequences and you can analyze their mutations. So now why does this matter for vaccine design? Well, it means that there are easy mutations and there are hard mutations. Easy mutations that readily occur in the germinal center and hard mutations or improbable mutations that are rare in the germinal center. And what you see here is a heat map showing all mutation probabilities for a single antibody's heavy chain, with amino acid positions going across the x-axis and each of the 20 amino acids going along the y-axis. And red and orange colors are improbable mutations. So what you can hopefully tell from this heat map is that there's a whole lot of red and orange. So the number of amino acid substitutions that are easily available to a B cell during its maturation are quite limited. We've used our Armadillo program to analyze HIV bNAbs, and we found that they are enriched with improbable mutations, many of which are critical for neutralization breadth. So these functional improbable mutations represent rate-limiting steps in bNAb development. And we've also seen that germline targeting priming immunogens don't sufficiently select for the combinations of these mutations that are required. So we really need a boosting strategy to get them. We use phylogenetic methods to reconstruct the maturation history of bNAb lineages, and that gives us a map of how to mature an antibody to neutralization breadth. But having a map can be really informative, but you could give me a map, point to where you want me to go, and I would still be completely lost without my phone telling me which way to turn. So we think that the immune system is a bit like that, and we may need to be more explicit and provide the immune system with turn-by-turn directions. And for that kind of navigation system, we think we need to increase the level of precision such that we're all the way down at the amino acid mutation level. So that's why we developed the mutation-guided vaccine design strategy, where we specifically design immunogens that can select for combinations of functional and probable mutations that are required for maturing breadth. So you can think of this as a GPS system for bNAb maturation. I'm going to show you results for applying this strategy to a lineage that targets the V3 glycan epitope. So real quick, the V3 glycan epitope is recognized by V3 bNAbs at the N332 glycan and a conserved motif at the base of the V3 loop. And the very best bNAbs to the V3 glycan site tend to recognize the epitope in a really highly similar way. Though their orientations can differ, they all reach their long CDR3s down to touch the base of the V3, and they squeeze past as well as interact with the N332 glycan. And the features that allow them to do this are also the features that make them hard to develop in infection or elicit with a vaccine. There's a low frequency of B cells in the naive repertoire with the CDR3 features that allow for broad recognition of the epitope. But in contrast to, say, CD4 binding site bNAbs, their maturation pathways are not as long, they're not as steep, with the number of improbable mutations being pretty modest compared to CD4 binding site bNAbs. So we think this is a really good target to go after with our mutation-guided vaccine strategy. So I'm going to show you results from applying that strategy to the V3 glycan bNAb lineage, which is called DH270. And the point here is to show you that we are learning the rules of how to induce bNAbs by guiding B cell maturation. Our entire V3 vaccine strategy does not revolve around trying to induce this one lineage in every vaccinee. Rather, our work on the DH270 lineage is really meant to establish how V3 glycan bNAb induction can be done. And then we can generalize out from there, applying the knowledge we gain on how to do that for one lineage to designing for whole classes of lineages targeting V3, and then for entirely different classes of lineages to other bNAb epitopes. Okay, so the way this works is first we computationally reconstructed the DH270 maturation pathway, and then we identified the hurdles along that pathway. And you can see that pathway on the left in the form of this clonal tree, and then we can annotate the improbable mutations onto that tree, which you can see in the red colors. We've also identified the minimal set of mutations that are needed to recapitulate the full DH270 breadth. And that number is just 12 mutations. So those 12 mutations are shown on the right, and they're color-coded by their probability. So reddish colors, again, being improbable. And you can see 10 out of the 12 mutations are improbable. These mutations become our targets for designing immunogens. And the idea is that if you can design immunogens that select for these mutations, we can direct the maturation process to where we want it to go, with our final destination being broad and potent bNAbs. I'm going to show you immunization data now in a special animal model made in collaboration with Fred Alt, in which we've knocked in the DH270 precursor. So the mice make the precursor B cells for the DH270 lineage, and that allows us to focus on trying to steer the evolution of those B cells to become bNAbs with our boosting immunogens. This has taken several iterations of regimens, and I'm going to show you results from the full gamut, where we go from prime only, where the goal is just to initiate the lineage, then prime and 1 boost, where the goal is to select for early functional improbable mutations, and then prime and 2 boost, where the goal is to select for this really very stubborn improbable mutation in the lineage called S27Y, that's a bottleneck. And then finally, prime and 3 boosts, where we aim to select for combinations of the critical early and late improbable mutations that help recognize signature sites in the epitope. We tested these immunized mice for neutralization in their serum. And with just the prime, we don't see any neutralization breadth against a panel of viruses sensitive to the DH270 bNAb. Those are the viruses that are shown at the bottom, going left to right. With 1 boost regimen, we get very little breadth. With the 2 boost regimen, now we start to see substantial neutralization breadth in the serum. And then finally, when we use the 3 boost regimen, we get robust neutralization breadth. And what we're really, really excited about with a 3 boost regimen is that we start to see neutralization of viruses that can only be neutralized in the late stage of the DH270 lineage, indicating we've been able to steer the response nearly all the way to the destination. We've performed a phase 1 discovery medicine clinical trial in humans, testing the first half of this regimen, where we primed and boosted with boost 1. We have data now emerging from after just the priming. And the middle panel shows characterization of the antibody response after priming, and we see robust targeting of the epitope, with 4.5% of the vaccine-induced antibody repertoire directed against the V3 glycan epitope. The right panel shows structural characterization of a handful of these antibodies, and they're in the V3 glycan bNAb orientation. We see this on-target response in 12 out of the 14 vaccinees. And the immunogenetics of these antibodies are diverse across all vaccinees, indicating we're getting a polyclonal response to the epitope. So we use the DH270 lineage as a template for designing immunogens to induce V3 glycan bNAbs, and when we vaccinate humans, we get a polyclonal response to the targeted epitope. So we think this shows that there's properties inherent in the priming immunogen that are generalizable, as it was designed for one lineage, but it works for many lineages. And we're really hopeful that the same generalizability will apply to the boosting immunogens as we get data back after the boosting, which is data that's coming soon. So in conclusion, by learning the rules governing the germinal center process, we've successfully employed a vaccine strategy to guide the B cell response along a targeted maturation pathway to elicit specific desired antibodies. And an immunogen designed to prime a specific lineage elicited a polyclonal V3 glycan bNAb precursor response in humans, suggesting that the key properties of the designed priming immunogen are generalizable for initiating diverse V3 glycan bNAb lineages. And with that, I'd like to thank the many, many people that make all of this work possible and the generous support of the funders. Thank you.
Thank you, Kevin. It's just a lot of hope there and a lot of exciting results, and very exciting to share the results of the clinical trial. Our next speaker is Kevin Saunders. And Dr. Saunders is a Norman Lettvin professor, distinguished professor in immunology and infectious diseases research at Duke and associate director of the Duke Human Vaccine Institute. And Kevin's now going to talk about the fourth target of broad neutralizing antibodies. That's the CD4 binding site, and success there, and as well mention clinical trials that are underway. Kevin, take it away.
Thank you, Bart. So just as a reminder, the CD4 binding site is this region that's targeted by broadly neutralizing antibodies that's highlighted here on the envelope in this cyan color. And this site is one that is targeted by broad neutralizing antibodies that are among the most broad and the most potent, making it one of the conserved sites that we'd like to target with the vaccine. So what we've done over the many years, starting back in 2013, is to study how CD4 binding site antibodies are developed. We use an individual living with HIV called CH505, where we can study how broad neutralizing antibodies have developed in this individual. We know that this individual developed 2 different types of broad neutralizing antibodies against the CD4 binding site. The first one called CH235. This antibody binds in a manner that's very similar to CD4, the receptor for the virus. and then CH-103, which binds in a different way, but is also equally effective. We've also studied the envelope response that was part of the viruses that co-evolved in the presence of these antibodies. We've been able to identify envelopes from this individual with either little modification or no modification that are able to bind to the CH-235 antibody clone. Importantly, we found an envelope that can bind to the precursor antibody. This is the antibody that we think can give rise to CH-235. And we can use this type of envelope as a way to try to bind and start off similar types of antibodies with vaccination. Similarly, for CH-103, we identified an envelope here shown in red that was also capable of binding to that inferred precursor that we think can give rise to CH-103-like antibodies. So together, these provide immunogens that can be tested to see if we can generate antibodies very similar to the ones that we found in the CH-505 individual. So I'll first start by describing some of the work that we've done using M5G458Y envelope and trying to generate CH-235-like antibodies. And so we first took that envelope and we moved into a non-human primate study where we immunized monkeys to see if we could generate antibodies that were very similar to CH-235. What we did is we took a bunch of CD4 binding site neutralizing antibodies and we saw the structures of them. We were able to identify an antibody that we call DH1285 that you can see here in this structural diagram where the envelope is shown here in this light gray and black, and then the antibody is shown here in the two different shades of blue. We could identify that it bound to the CD4 binding site. But to really know if this antibody was like CH-235, we needed to really look at the atomic level detail. So when you zoom in and look at exactly how this antibody that comes from the vaccinated monkey, how it binds to the CD4 binding site, you can see several key features. So first, you can see this green portion of the antibody contacts the red CD4 binding loop. You can also see that there is an arginine at position 71 in the antibody that forms this contact with aspartic acid at 368 in the envelope. Now that is important because we know this binding mode is the exact same binding mode that all of the known human broad neutralizing antibodies that mimic CD4, this is the binding mode that they use. As an example, you can see CH-235, which was our target for the vaccine. You can see it again uses the same green portion of its antibody to contact the CD4 binding loop, and it also uses the same arginine at position 71 to make this contact with aspartic acid at 368. So not only were we finding CD4 binding site neutralizing antibodies, but they are binding in the precise way that we know human broad neutralizing antibodies actually target the envelope. And so this was able to show that the immunogen that we designed is able to initiate antibodies that look very similar to the CH-235 antibody. We next wanted to see could we actually evolve these antibodies to develop broad neutralization. So to do this, what we use is a humanized mouse model where the mouse actually has the genes introduced for the CH-235 precursor. So the mouse makes antibodies that look like the precursor that gives rise to CH-235, as well as its own endogenous mouse antibodies. We then can use 3 different envelopes and vaccinate these mice to see if we can help guide the affinity maturation or evolution of the antibody to hopefully develop broad neutralization. So shown here are just 3 examples of monoclonal antibodies that were made by these mice after they were immunized with the 3 different envelopes. You can see here different viruses going across the top, shown here. And then these are color-coded based on how potently they neutralize. So the darker the red, the more potent the neutralization, the lighter the color, the weaker the neutralization. The starting place, this precursor antibody that we think can give rise to CH-235, you see can only neutralize the virus that matches that M5G458Y envelope that we know can interact with the precursor. It doesn't have activity against any of these other isolates. After we vaccinated the mice, although their antibodies start out as this CH-235 UCA, they're able to evolve now to recognize and neutralize all these different other isolates of HIV. So the ultimate goal here is to move the antibody from the starting place out to a broad neutralizing antibody. And these antibodies show you that we are able to do that to a certain extent using these 3 envelopes. We next moved into the humans to see if we can actually recapitulate some of these successful results that we've seen in animal models. To be able to do these studies really quickly, we're encoding the HIV envelope as an mRNA. And this allows us to manufacture the vaccine very quickly and then move it into discovery medicine trials rapidly so that we can get answers to the question. The humans received 2 immunizations, as you can see here. And then we isolated serum to look at the CD4 binding site neutralizing response. So to characterize the neutralizing antibody response, we used two specific viruses that we engineered to tell us whether or not we're generating CH235-like antibodies. So just to describe what these viruses are, you can see here we designed one virus that's very sensitive to the broad neutralizing antibody precursor. It can neutralize it fairly well. We also designed a very similar virus, but it has mutations introduced that now make it resistant to the CD4 binding site broad neutralizing antibody precursor. And you can see neutralization has now dropped. So we look for this same pattern in our vaccinated participants in the study. And as you can see here, they have serum antibodies that neutralize the sensitive virus. But in this virus, where we've now made it resistant to CH235-like antibodies, you can see the neutralization drops at different extents in each of these participants. And so what we're seeing here is that the serum antibody response recapitulates the neutralization profile of the CH235-like precursor, suggesting that we are indeed generating antibodies that are targeting the same site. We've next moved to look at whether or not we can do a similar type of approach and generate CH103-like antibodies. So in these studies, we used the transmitted founder envelope, or TF envelope, that's represented as the red envelope. We did this study in humans again in the HVTN 300 phase 1 trial, where we've immunized people 4 times using 300 micrograms of this protein mixed with adjuvant. And we're looking to see, are we generating CD4 binding site antibodies that are similar to CH103? Again, we were able to isolate monoclonal antibodies from this for the vaccinated individuals. And we used these 2 different viruses. So one virus that's sensitive to the broad neutralizing antibody precursor, and then one virus that's resistant. As you can see, each of these antibodies lose neutralization activity when you test them against this resistant virus that's been designed to tell us whether or not CH103-like antibodies are present. To more definitively show that these antibodies are like CH103, we again use structural biology to try to understand how the antibodies bind to the envelope. And when we zoom in and actually look at the interaction of how our vaccine-induced antibodies bind to the envelope, we can then compare that to CH103. So CH103 structure binding to the envelope is shown here in this ribbon structure. An envelope is shown here in gray. And then the smooth surface that you can see shown here, it represents how the vaccine-induced antibodies bind. As you can hopefully appreciate, that you can see that they overlay quite well, showing that the vaccine-induced antibody binds to the envelope, the CD4 binding site epitope on envelope in the exact same manner as CH103. So overall, what I've been able to show you today is that we've been able to induce CD4 binding site neutralizing antibodies in humans, monkeys, and mice. The antibodies have the correct genetics for CH235. They have CD4 binding site dependent neutralization, and they bind structurally with the correct binding orientation. And this tells us that envelopes and antibodies from people living with HIV-1 can inform and enable vaccine design. We have successfully designed immunogens that bind to the precursor antibodies and can give rise to CD4 binding site broadly neutralizing antibodies. We are eliciting CD4 binding site broadly neutralizing antibody precursors in humans, monkeys, and humanized mice. And then lastly, the sequential vaccine in humanized mice can drive antibody maturation, resulting in neutralization breadth. So I'm just one representative of a large team that works on this project, both from the HVTN that performed the human clinical trials, and as well as all the discovery science that goes on at Duke, and also with our collaborators at Harvard. I always like to thank the funding sources, and I'll hand it back to you, Bart.
Thank you, Kevin. Just beautiful studies and very exciting. And again, emphasizing the translational ability of this entire team and the partnerships that the team enjoys. Our last speaker today is Dr. Lindsay Baden. Dr. Baden is at Harvard and is the Vice President of Clinical Research at Mass General Brigham, and he's the Naval Family Professor of Medicine at Harvard Medical School. He's also a member of the HIV Vaccine Trials Network that performs all these terrific trials. Today, Lindsay's going to talk more about why we need an HIV vaccine and go into some more details of clinical studies for bNAb development, talking about clinical studies not only by our team, but also others in the HIV vaccine field to give a complete picture of the progress that's being made. Lindsay, please take it away.
Thank you, Bart. And thank you all for joining. I'd like to bring things together as to why we need a vaccine and how we are in hot pursuit of developing one. So as we all know, there are more than 30 million people globally infected with HIV, and only a fraction have access to treatment. And despite new therapies and new prevention modalities such as PrEP, there still are an unacceptably high number of new infections, with current estimates, or at least from 2 years ago when data are available, with 1.3 million new infections per year. As illustrated on this image, with our new prevention modalities, we've been able to decrease new infections, but not nearly at the rate that we need to if we want to control this epidemic and create an HIV-free future for our global community. As this image demonstrates, and this image is trying to help highlight the fact that most or nearly half of the new infections per year are individuals who do not identify as high risk. So if we take a hypothetical 1,000 individuals and 20% of them may identify as high risk, new infections may be 6 out of the 200 individuals and 4 out of 800 individuals, raising the concept of number needed to treat to prevent a new infection. In those who self-identify at high risk, that number may be relatively small or manageable, while those who do not see themselves at high risk, that number needed to treat, in this example, 4 out of 800 or 200 to prevent 1, and in populations who have limited access or see themselves as even lower risk, the number needed to treat may be even higher, further highlighting the point that Dr. Haynes made earlier, that we've never been able to treat ourselves out of an epidemic, particularly one that affects all of us. We must develop a vaccine which is scalable globally in an equitable and highly deliverable manner, as currently being done with the many globally delivered vaccines that all of us, I hope, have received. This image is highlighting the 11 trials or large field trials that have been done in pursuit of an HIV vaccine with 7 different concepts. And of them, the RV144 demonstrated modest efficacy at 30%, allowing us to explore and understand potential correlates of protection and help guide the science that we want to develop protective immune responses. And then the AM study, which is passive immunotherapy, which is delivering the VRC01 or the anti-CD4 binding site antibody, which Kevin just discussed in the previous talk. And I'll come back to this study in a moment. I want to highlight key concepts that have been raised, given that these are very challenging concepts, the issue of eliciting broadly neutralizing antibody. And in this important publication over a decade ago, in an individual, and several individuals, but in a particular individual who's a long-term non-progressor, who's infected with HIV, one is infected with HIV, develops an immune response, an antibody response. The virus escapes, and a new antibody response is developed, and the virus escapes, and a new antibody, and so on and so on. And through this chronic long-term infection in a small number of individuals, they're able to develop a broadly neutralizing antibody that is able to identify and neutralize HIV and the kind of HIV they are infected with. This concept, this set of observations was critically important to us as a field to understand and establish the concept of broadly neutralizing antibodies, which are rare and come from improbable mutations, as Kevin mentioned earlier in his talk, that can allow the neutralization of HIV and diverse HIV strains that circulate globally, thus allowing us to pursue the development of these kinds of antibodies at key targets that are conserved in HIV and, if neutralized, can block transmission. This image, which Bart presented at his opening, but I wanted to come back and really highlight because it builds on all of the talks thus far, is in order to develop that broadly neutralizing antibody that can recognize the global diversity of HIV that an individual may be exposed to, we must make sure that the appropriate B-cell lineage is engaged, a lineage that may be reluctant to emerge or may need special targeting to emerge. And then we need to help that lineage mature with the appropriate additional immunogens that allow it to develop down a certain path so it has broadly neutralizing properties. None of these antibodies may be broadly neutralizing. So thinking of them as broadly neutralizing out of the starting gate is not the way to think about it, but rather it has the potential to become broadly neutralizing. And if given the appropriate directions, the correct GPS, we can help it mature so eventually it matures to the fully broadly neutralizing antibody. This requires iterative trials that can demonstrate the elicitation of the right lineage and can demonstrate proof of maturation of that lineage, where the subsequent immunogens, boost number 1, boost number 2, boost number 3, are predicated on the prior immune elicitation that is developing down the appropriate track. These are small, rapid studies that can allow proof of principle, proof of concept that this can be done so that we ultimately land with our broadly neutralizing antibody that is able to be elicited in large parts of the population. And these are what we call the HVTN, HIV Vaccine Trials Network, and NIAID, NIH-funded network to develop an HIV vaccine. And we have a discovery medicine series of trials so that we can build on the preclinical work to rapidly assess safety, immunogenicity, and proof of principle in the human biology so this can be extended to where we ultimately want to go. So there are proof of principle. In the AMP trial, antibody-mediated protection, this was a study done globally, as noted down here, in many sites around the world, in the Americas, in Europe, in Africa. And this trial was given VRC01, which is a CD4 binding site antibody, and it was given as passive immunotherapy, so like IVIG, intravenous immune globulin, which we give to our patients globally every day in the hundreds of thousands of doses as it treats many different conditions, including antibody deficiency. We can give this monoclonal antibody that targets a vulnerable site of HIV. And in doing this, we were able to show that if given passive immunotherapy, so the antibody alone, if individuals were exposed to a susceptible virus, to an HIV virus that was susceptible to this antibody, there was high level of protection. In HIV viruses that were less susceptible or resistant to this antibody, there was breakthrough infection. This study, though not what we wanted in the sense of a globally actionable antibody, was incredibly important to the field, to the science, and to our development in that it was proof of principle that we can give immunotherapy or IVIG globally. We can give it and demonstrate that it can block acquisition, meaning proof of principle that if you have circulating antibody active against the HIV strain you're exposed to, it can block acquisition. Critically important to advance the field and fundamental proof of principle. So highlighting, bringing together the earlier talks and thinking about how we can go forward to have an HIV vaccine that's protective. If we think about the HIV spike, as discussed before, we can have sites of vulnerability at the membrane proximal region, at the CD4 binding site, at the V2 glycan, the V3 glycan, the fusion peptide. Having identified from fundamental science with confirmation in preclinical studies that these are sites of high conservation in HIV needed for its successful replication. That then allows us to target them so that we can develop antibodies that can target these highly conserved sites needed for HIV's success and allow us to come up with a multi antibody cocktail that can get around an individual kind of resistance. So as Dr. Haynes said at the beginning, if we have 3 antibodies, one from at least 3 different epitopes, the likelihood of an HIV virus being resistant to all of them is extremely low. And this can then become a cocktail or either through passive antibody, but more importantly through vaccine elicitation that can give durable immunity theoretically to individuals who are able to make multiple bNAbs at high concentration. What this image shows, and it's bringing together the prior talks and also looking forward, if we think of these 5 sites of vulnerability, these are multiple trials that have either been completed or are currently in the field. Over a dozen have been completed or in the field attacking these different sites of vulnerability, and another dozen are on the horizon where we can develop proof of principle that we can elicit the correct lineage and we can help it mature to have the appropriate broadly neutralizing properties that make it attractive as a vaccine or combination vaccine to be able to bring out this immunity in all of us, which is really the hope, so that we all are protected. So I find this incredibly exciting that the science has brought us to a point where we understand sites of vulnerability, we're able to do the foundational science to develop the immunogens, and we are now in multiple clinical trials being able to demonstrate the elicitation of these types of likely protective immune responses once we're able to bring them to full maturation with the different booster concepts that have been raised already. So as noted, this work really is a community activity with multiple sponsors who have been critical, multiple investigators across many institutions, as well as Duke, the HVTN, which is coordinating many, if not all of these studies, and really appreciate the contribution of all of our volunteers who are willing to work with us so that we are able to demonstrate the human biology that we think will be protective and therefore accelerate our ability to develop these vaccines. So thank you, and let me turn it to Dr. Haynes.
Thank you, Lindsay. That's terrific. If you can, yeah, and I just want to thank you for that terrific overview. And I'll just briefly sum up here and talk just for a second about all of our partners. This vaccine is going to be made and successfully deployed by a global cooperative community. And this slide just shows some of the wonderful partners that we've had over the years with our HIV vaccine group and the payers, and the success that we have had and others in the field have had that Lindsay gave a very nice global overview is a testimony to the steadfast support of the payers, the NIH, the Bill and Melinda Gates Foundation, other federal funders, USAID, and Eurovac, IAVI, MHRP, folks and groups all over the world that are working hard to end the pandemic. And so our thanks, and what an honor to be involved with all of you that we've had the pleasure of working with over the last number of years. We hope that today we have had a chance to show you some progress and also to show you how difficult this vaccine is to make and how difficult the science problem is that we've had to solve. There is a reason that 20 years after the full-scale funding by the NIH and the Gates Foundation for the Global HIV Vaccine Enterprise, that we still don't have a vaccine. But the message today is that we have promise and we have progress. and it's a pleasure and honor to share that with you. This is the first vaccine that we've had to understand the very, very basic host-virus interactions in order to make this vaccine. It's learning the rules, as several people have said, is very key to understanding how the immune system works. If we can solve this problem, we'll be able to solve many more other problems where immune system regulation is going to be necessary, such as autoimmune disease, etc. So I'll stop there, and we'll ask for questions to come forward. And we'll be happy over the next 15 minutes or so to answer any questions.
Thank
Asli,
you, Bart.
do you want to say anything?
Yes, I wanted to thank you all for the really exciting presentations, very informative. We do have quite a busy question and answer inbox, so I'm just gonna shout out some of the questions. I'll go roughly by order because I don't know how to sort them by relevance. There's different topics. So one of them is, it will take time to develop the right bNAb response, during which people will remain susceptible to HIV acquisition. So assuming a successful regimen is developed, how do you envision the rollout? How can we ensure that people remain protected from HIV during the rollout phase?
It's a great question, and I'll make a comment and then ask for others to comment, particularly Lindsay. One of the things we found in one of the early HIV vaccine trials that we were very worried about was that because, as George Shaw said, that it takes people who are living with HIV many, many years to develop broad neutralizing antibodies, are we going to have to vaccinate for many, many years? And at least in our first clinical trial called HV-103, we were only able to vaccinate for about 6 months, but we got these rare and probable mutations, and we got heterologous neutralizing antibodies in a much shorter period of time than would be expected from a person living with HIV. So the hope is that the time, or the window of time, will be shorter than what happens in people living with HIV. Secondly, I suspect that as the vaccine is rolled out, companies and the clinical trials units will also be rolling out assays for determining, have the so-called correlates of immunity, as Beatrice Hahn was talking about, getting to a particular level of a serum neutralizing antibody will be obtained. So we'll know what that window is, and we'll know if people should be protected or not. Lindsay, do you have any addition to that answer?
2 additional concepts. We will always want to maximize HIV prevention. So if one is unvaccinated, one should minimize risk. If one is fully vaccinated, one should minimize risk. So until we know that we can stop transmission, we will need to step on the gas in all ways to stop transmission, which also includes treating everyone, diagnosing everyone infected and treating them so that we can bring the community viral load down. The other concept in terms of the maturation of the B cell, our current experiments are a bit clumsy because we are doing the science to figure out what's possible. Once we know what's possible, one could imagine depot products that could time release different immunogens, so won't require dozens of vaccinations in short periods of time. Once we figure out the science, as Bart says, then there are many technological advances that could allow us to give an immunization that actually is releasing the appropriate sequence of antigen over time through a depot or other process. So I think we'll be able to simplify once we get it right. Over.
Asli, back to you.
Thank you. So then there's 3 questions which kind of feed into each other. The first says that at R4P it was suggested that there is a potential for steric hindrance when using a cocktail of bNAbs. Are there any specific concern? Now, following up on that, there is another question saying, is there current thinking on how these 4 bNAb responses would be combined in a vaccine regimen and how that would look like, separate shots, 2-shot vaccine regimen? And then a third one saying the challenge of getting a durable and effective response.
Right. I'll turn the first part of it for combining these maybe to Kevin Saunders. We're working together to try to combine different immunogens. The group at Scripps is working together to combine immunogens. And I think the general notion, Kevin, is that if they're against different epitopes, different binding sites, there's not, so far, we've not seen any interference. Your thoughts on this? We'll come to durability in a minute.
I think there's basically two approaches to this. So one is you mix immunogens together, and then you're targeting different types of bNAb responses with each immunogen. And I think in some work that I've seen and some work that we've done, it seems like as long as you're targeting these different sites with immunogens that are pretty adept at binding to those, it seems that that's possible to do where you mix them together and you don't get kind of cross-competing or interference with those. In one of the studies that we did where we were using V2-Apex targeting immunogens, we also had those animals that were being immunized for MPER. And so they're able to generate MPER antibodies and they're generating V2-Apex antibodies at the same time. And doesn't seem to be really any major interference that we know of as of yet. And then we know there's others that are doing similar studies. The other thing that we're doing, though, is actually taking the epitopes that we know are critical for starting off certain broad neutralizing antibodies and combining them into one envelope. And so that's where we'll really find out, is there steric hindrance and is there any interference? When you put those together into one envelope, will we be able to generate the same 3 or 4 different broad neutralizing antibody classes that we're going after? And those studies are just starting, so we don't have the final data for that yet. But at least in vivo or in vitro, when we do binding assays, it seems like this is still a plausible approach.
Thank you. With regard to durability, that's a terrific question. And this is a different vaccine than measles, mumps, rubella, polio, and what have you, because in those vaccines, you can rely upon the secondary immune response. And the person may be infected, but they'll never know it because the immune response comes up so fast that it eliminates the infection. Whereas with HIV, it infects us and gets into our genetic material within probably the first 3 days after transmission. And so we don't have time to develop a new immune response. We've got to have a high level of durable antibodies and T cells. And we didn't have time today to talk about other immune responses, but we're working on a lot of different kinds of immune responses to help the antibodies. And so that involves studies on inducing a kind of cell called a long-lived plasma cell that lives in the bone marrow. And there's a whole team both in the Duke CHOV-D and in the Scripps CHOV-D, as well as in other groups around the world that are working on the biology of long-lived plasma cells. And we're very hopeful that that problem is going to be solved in tandem so that when we have the immunogens designed correctly, they can be formulated in order to induce very broad and persistently potent neutralizing antibodies. So that's a big deal for us. And thank you for that question.
Thank you. Mitchell is asking how a potential target product profile would look like of an HIV vaccine, especially in light of now long-acting prevention options with once a year a shot. Of course, the vaccine will still be needed, but the context would be different. So is there any thought about the target profile?
Yeah, so the brief target product profile gets to the correlate of protection. We have to have the induction of what's called an inhibitory dose 80 of around 1 to 100, 1 to 200. Beatrice mentioned an inhibitory dose 50 of around 360 to 400. So we know we've got to be there in that area, hopefully higher. The issue of interfacing a vaccine with long-acting PrEP. I think Lindsay really touched on this. We've got to be preventing HIV in every way that we can and in every individual. Lindsay, you want to comment on sort of how that might interface with the vaccine?
I think it's a terrific question. And as already alluded to, what we would want to do as a community is to maximize prevention. And one could imagine a long-acting PrEP that could be used as cover while the immune response is elicited. Because the sustainability of long-acting PrEP, even at a couple of times a year or once a year, in all parts of the world may be challenging. So one could imagine, depending upon the data we have as to the optimal use of PrEP or other kinds of prevention strategies, how to develop the immune response from a vaccine while we maximize prevention with the idea being that a vaccine would be long-term sustainable and economically favorable given the cost of delivery. Once you have the immune response, as we see with the EPI program globally, that immune response can last years to decades, which would be our intent. So I see this as terrific synergy.
Thank you, Lindsay. And then Mitchell, and I don't think only Mitchell, I think a lot of us are wondering if you can share thoughts how the current budget cuts might affect the pace of the research. And feeding into this, another question, what role can advocates play around the world to ensure that the research continues?
Well, certainly advocates around the world can advocate and share their enthusiasm for the importance of the work to their own governments and to the payers, and thanks to the payers for their sustained support, and with high hopes that the support will be continued. It's taken a global effort and a sustained effort over the past almost now 30 years, 20 years of the HIV program, to make the kind of progress that has only been realized in the last 2 years, and gave rise to this particular webinar and the excitement that the field has recently generated. So we are in high hopes that the work can continue.
Does anyone else want to add some thoughts?
Any other comments? Lindsay?
Happy, and then others. I think that resources is time. So the more resources that are available, it can fuel this, so that all of the work that's going on, which is a tremendous amount of science has been presented in the last hour, made accessible, interpretable, and almost looking simple. But the amount of complexity and depth here is tremendous, and money equals time. So the more investment we have into these preventative modalities, the sooner we will have them. And I would, in my comments earlier, with at least 1 million new infections a year, if there is access to ART, antiretroviral therapy, is made more difficult, that may increase the number of infections, increasing the community viral load, if I can use that concept, which may increase transmission. So the best way for us to be able to solve this is to have the resources to do the science as quickly as possible, so we actually get to the answers that we all want. So to me, money is time.
Ashley?
Thank you. I guess there is not much to add. We have a few more questions coming back to the basic science. I'm just gonna take one of them now due to time constraints. And that is if the bNAbs would be able to prevent the establishment of a viral reservoir, or if a T-cell response would also be needed.
Who would like to take that? Well, I can certainly comment on it, given the work that we're involved with the Cure/AIDS Martin Delaney centers. We believe that an effective preventive vaccine that can induce broad neutralizing antibodies is going to have an effect on being one of many different modalities that might impact decreasing and eliminating the latent pool. A number of studies have shown that when broadly neutralizing antibodies are given to people living with HIV and on antiretroviral treatment, or ART, that when the ART is removed, that the persons living with HIV can control their virus in certain conditions, in certain states, and in certain individuals. And so there is hope that combining broad neutralizing antibodies passively administered, along with things that can reawaken the latent pool and make them more vulnerable to the immune system, can combine to help eliminate the latent pool and achieve the Cure/AIDS status. We believe a preventive vaccine, once it's successfully made, was going to be an important part of that kind of treatment. And we're already doing studies in non-human primates of vaccinating with George Shaw, HIV-infected macaques to try to control the viral load. And those studies look promising.
Thank you, Bart. Thank you, everyone, our participants, our presenters. It was informative. It was exciting. I think it makes a lot of hope. And let's continue the enthusiasm and let's make sure that research continues. Again, thank you all. I want to inform everyone that the recording of this webinar will be made available on IIS Plus, and please keep engaged and have a lovely weekend.
Thank you all.
Thank you.